The overall goal of this experiment is to reconstruct a 3D inflammatory gingiva tissue model by using 3 types of human cells in vitro.
This method can help study wound healing, tissue regeneration, inflammation, and screen potential medicines for periodontal diseases. The main advantage of this technique is that this is the first method to build an in vitro model of inflammatory gingiva using human cells with a 3-dimensional architecture.
In a 15 ml sterile tube, add collagen type 1A, 10% concentrated MEM-alpha, and 10% reconstruction buffer. Then leave the collagen mixture on ice till the next use. Next, position the 24-well culture inserts with 3 micrometer pore size in a 24-well culture plate. Then use 0.25% trypsin EDTA solution to detach the HGFs from the culture flask. Next, suspend the detached cells in 10 ml of culture medium. Then count the number of cells using a Burger Turk cell counter. After isolating the desired cell number, centrifuge the cells at 190 times G for 4 minutes. Then suspend the cell pellet with the collagen solution and leave the tube on ice. Next, add 0.5 ml of cell collagen mixture into the culture insert without producing any air bubbles.